So as we know, organisms with common ancestors share common traits. And we can use these common traits to assess the course of evolution. Right? And so over time, organisms build up these new traits, and they tend to keep those traits. Right? Occasionally, we will lose a trait because it is disadvantageous to have it, but more often than not, the traits are conserved. Right? And so we can kind of say who came soonest in the course of evolution, who came earliest, by who has the fewest traits. Right? And this is most effective when we're looking at a linear relationship, a linear sense of common ancestry. And so it's not just like a raw, who has more traits, a spider or a human. Right? You have to be looking at like who has more of these specific traits along this course of evolution that we've already kind of plotted out. And so when we plot out these relationships by traits, okay, we call it a cladogram. And we use these cladograms to depict evolutionary relationships. Right? And so here is an image of a very simple cladogram. This is a one-clade cladogram. You can also branch off the cladogram in other spots, and you have multi-clade cladograms. Right? So at lizard, I could also have another line that goes to like crocodiles, and another line that goes to snakes. As I could build a clade off of this clade. Right? So I could go up here and build another clade. I can do that. Okay? But looking at this simple cladogram that we have in front of us. And so the cladogram is built from left to right. And as we move from left to right, we add traits to the storyline. So anything that is to the right of the trait listed has that trait. In this particular cladogram, anything to the right of lungs has lungs. So the salamander has lungs, the lizard has lungs, the hamster has lungs, and the chimpanzee has lungs. Anything to the left of that trait does not have that trait, and therefore the salmon does not have lungs. Okay? And it goes upwards and onwards, right? And so in this line of common ancestry, we assume that the salmon came first, because everything after the salmon has lungs, everything before the salmon will not have lungs. Okay? If we look at claws and nails, right, everything to the right has claws. So lizards have claws, hamsters have claws, and chimpanzees have claws. Salamanders and salmons do not have claws. Right? And so claws showed up right here, and because we keep traits, we tend to keep them. Everybody after they show up still has them. Right? So that's how we structure cladograms. Typically when you are looking at a cladogram, you are also going to see this kind of accompanying chart that has the traits marked off. Right? So this is the same information presented differently. So who has lungs, right? The salmon did not have lungs, no check. The salamander, the lizard, the hamster, and the chimpanzee all had lungs. Check, 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 check. Okay? Who had claws? Okay? The salmon and the salamander did not have claws. The lizard, the hamster, and the chimpanzee all had claws. And so forth and so on. And so we use these to try to rough out who is related to who in an evolutionary timescale. And who showed up first as well. Right? Nowadays, we can use gene similarities with actual numbers to create these cladograms rather than, oh yeah, that guy has lungs and that guy doesn't. Right? And determining traits can be kind of subjective. Right? Like what you call a lung might be something different from what I call a lung. And so like maybe salmons do have lungs because they have gills. And so maybe I think gills are lungs. They're not, but I could think that. And so I would put salmons after lungs. There's subjectivity there. Whereas genes and DNA sequences have very specific numbers, right? And so if I have 36 of the same genes, okay, I put that before having 39 of the same genes. That's a number. There's nothing subjective about numbers, right? So we can build cladograms using genetic sequences as well, which is better. Yeah, cladograms. Bye.